Photoreceptors and light. Light enters the eye and hits this photoreceptor. It passes straight through the optic nerve, the bipolar neuron, to the photoreceptor. At the photoreceptor, the light is absorbed by the light-sensitive pigments up here. When the light reaches those sensitive pigments, it bleaches it, causing a chemical change. This triggers a nerve impulse along the bipolar neuron to the optic nerve, straight through the brain. Humans have two types of receptors, photoreceptors. We have rods and we have cones. Rods are found in the peripheral parts of the retina, and they give information in black and white. Whereas cones, they're packed together in the fovea, and there are three types: the red sensitive type, the green sensitive type, and the blue sensitive type. So, what happens is the light rays go straight through, passing all of these into the photoreceptors. It bleaches the light sensitive pigments, causing a chemical change, and the direction of the impulse goes straight down that way. So let's have a look at the photoreceptor rods. Rods contain a light-sensitive pigment known as rhodopsin, which is made up of two chemicals known as retinol and opsin. So what happens to rods in the dark? In the dark, rods are not stimulated. The sodium ions are pumped out of the cell using active transport, but they can diffuse back into the cell through open sodium channels. This makes the inside of the cell very slightly negative, causing the cell membrane to be depolarized. What is depolarization? Depolarized just means there's not much difference in the charge across the membrane. Now, this very slight difference in cell membrane charge triggers the release of an inhibitory neurotransmitter. So we have an inhibitory neurotransmitter being released. This neurotransmitter then inhibits the bipolar neuron, and it prevents it from firing action potentials, and so no information is sent to the brain. So what happens in the light? In the light, the rods are stimulated. When the light energy reaches rhodopsin, it breaks the rhodopsin into retinol and opsin, and this is known as bleaching. So when light hits the rods, they are stimulated and bleaching occurs. This bleaching causes the sodium channels to close, and so the sodium ions are moved out of the cell but cannot diffuse back in. So they start building up on the outside. Once they build up on the outside, the inside of the membrane is much more negative. This causes the cell membrane to be hyperpolarized. So there is a difference in charge. When the rod cell is hyperpolarized, it stops releasing the inhibitory neurotransmitters. So the neurotransmitters are not released. This means there is no inhibition of the bipolar neuron. So the bipolar neuron can start working again. The action potentials are sent across the bipolar neuron, and they're also sent along the optic nerve to the brain. If the potential difference reaches the threshold, the action potentials are released and they're transmitted to the brain via the optic nerve.